Hi, everyone. Hi. My name is Kirby Wallace, and I'm a doctoral student in the Biomedical Sciences program at the University of Tennessee's Health Science Center. And I do all of my laboratory work at St. Jude Children's Research Hospital. Tonight, I'm going to be debunking a few myths about CRISPR, and I'm going to give you a sneak peek into the work that I do with gene editing technology every day. But first, I have a couple of questions for everyone. So, how many of you have heard of CRISPR? All right, a few. How many of you have failed your introductory to biology course? Okay, more of you. All right. Well, don't worry too much about the technicalities of this next slide, but I want to get everyone on the same page. Cells have DNA, and DNA is composed of thousands of genes. Scientists can use CRISPR as a gene editing technology that will specifically recognize genes and cut them out of DNA. This can be done for one gene in a cell or for multiple genes in the same cell. All right, I think I've got everyone caught up. Hopefully that worked for you all. But why is this important? I'm encountering CRISPR not only in my lab, but also in the news. It's a hot topic in the media right now, but not everything that you hear is true. CRISPR is being used to study diseases from HIV to sickle cell, Alzheimer's, and cancer. But it's also being used in food production and agriculture. So I want to talk to you about a couple of myths that keep coming up when I hear descriptions about CRISPR. I hear that CRISPR is fast, that it's easy, and it's cheap. This is just not true. Sure, it's fast if you understand all of the techniques that you need in order to perform experiments. It's also easy if you have all of the understanding of biology that you need to get your work done. Also, the ability to ask very interesting and important questions, and then to analyze your data. And last, sure, CRISPR is cheap. When you have a state-of-the-art lab that's equipped with all of the machinery and reagents that you need in order to get your experiments done. So I think saying CRISPR is fast, cheap, and easy is a misrepresentation of why scientists are excited about this technology. We're excited because CRISPR is a powerful tool that streamlines the way that we study disease and biology. So now that I've told you a little bit about some of the myths in the field of CRISPR, and hopefully have debunked them, I'd also like to talk to you about some of my work in the lab and some exciting results that I've gotten using CRISPR technology to edit the cells that I work on. So I work on a pediatric-only cancer called neuroblastoma. And neuroblastoma arises in the sympathetic nervous system, or the fight or flight nervous system. Neuroblastoma occurs when cells that would normally form the nervous system have genetic changes that cause these cells to form cancer instead. So I use cell imaging to study neurons. Neurons are the type of cell that form the nervous system. And in typical conditions, when I add media to normal cells, or a liquid that tells the cells to become neurons, they form beautiful neurons. They're very healthy with lots of networks. But this is where it gets interesting. When I use CRISPR technology to remove a gene that I'm interested in from normal cells, and then add the same conditions, the same liquid, these cells no longer form neurons. They're not healthy, they don't have the beautiful networks that we expect. So what that tells us is that removing one gene, this one genetic change, prevents the formation of neurons and therefore prevents the formation of a healthy nervous system. So my hypothesis is that then these cells with this genetic change take a step toward becoming cancer. With this information, we can now find ways to develop new therapies that can make these cells become neurons, or we can use drugs that kill these cells so that normal cells will take their place. The ultimate goal of my experiments is to find new treatments that will save lives. And this is why I love using CRISPR to edit genes. Thank you.
You already know. Damn, girl, how you get all of that? And them jeans, how you get all of that?